Hello, Dr. Eve Pearce here. I'm so sorry that I couldn't be at your conference on Saturday, the 1st of July at the AOMT. I was really looking forward to seeing you and giving this presentation as an introduction to nutritional genomics, and we were going to specifically explore some inflammation. So what I thought I'd just do is give you a little recording so that you have a good taster of this area that hopefully you'll be excited to try and look into. So there's a couple of slides here about my experience and where I've come from. So feel free to read through those and my bio, but we'll be starting this webinar with just an overview of where we're going today. So we're going to just look at human genetic variation and modern genetics and just get a flavour for the whole area. Then I believe that you had a lecture around functional medicine and this exciting area of integrated medicine. So I'm just going to briefly go through how our nutritional genomic testing really fits into that model. It'll be a brief look through molecular biology, a bit of 101. It'll be a bit of a revision for many of you. And then we'll focus a bit more in on what is actually nutritional genomics and look at some terminology. Then I'm going to move on to epigenetics, which is an area of research that's up and coming and very interesting. And bringing all of this together, we'll be looking at the myDNAhealth approach, how that works in practice. At the end, I've put in a inflammatory risk analysis framework, which is something that we use with our practitioners in the nutrition side to help them present this information to a client or a patient with just a couple of example polymorphisms or genetic variants there to give you a flavour. So the idea of this whole talk is really to inspire and interest you in this area and hope that you look into it a bit further and become more confident with using it in practice. So really, if we think about understanding human genetic variation, we look at some facts here. We're a relatively young species. From sort of an evolution point of view, we haven't been around that long. And that hasn't really given us that much time to produce many variations. So essentially, we're all very much the same, but we can see physically that there are a lot of differences. And that's really where we're looking at these differences physically. And so you may have or may have not have heard of the Human Genome Project, but we know now that we have about 3 million base pairs of DNA. And actually, there was a lot of information that's come out from that, and we thought that that would be the end of the story and we would have the whole map as such of us, but now we're beginning to realise that there's much more to the story and it's much more complicated than that. So just some basics. No two people are identical, except obviously monozygotic twins, so identical twins. But even they occasionally do have some genetic and epigenetic differences. So we'll talk about that as we go through. And this occurs throughout development. Any two people can have about 10 million base pairs, which are different. Some of the research is a bit vague on this, but we think it's about 0.1 to maybe 0.3% of our entire genome, which doesn't seem like a lot, but actually is the bit that contributes to our difference from everybody else. So essentially we're all very much the same, but there's this very small fraction which is different and leads to our genetic variants. And so we know that around the world, all populations of humans are essentially the same, and the differences lie in the individual. So that's where we're coming from here today. So we're thinking about the individual person, patient, client, whatever they may be. We're thinking about that person and trying to tailor their possible genetic variants or their epigenetics to themselves. So just to expand on the Human Genome Project a little bit, at the time it really revolutionised genetics research, and this is when we were literally looking at the genetics. We were looking at that base pair code. So if you remember, we've got this base pair pairing of A to T and C to G. It was officially completed in 2003. 25th of April is officially DNA Day. They started on that day and finished that day. There is a website that you can go to, just Google it. There's the logo of the Human Genome Project there in the corner for you. And on that website, they do occasionally post updates because there is continuing to be a small amount of research going on under this project. The project was the whole world got involved. The USA really led and collaborated the project together, but there was contributions from the whole world towards this sequencing of one particular person. So from this, we've identified the human DNA sequence, which is amazing to have and is a great resource, is available online. And it really highlighted approximately 30,000 genes, which is our genome. So our genome is the global manifestation of our genetics. We were a little bit disappointed with this because there's, other wheat, for instance, has maybe 60,000 genes. So we are pretty low in the gene stakes. So that's when we started to understand that there was something more going on with us rather than just our genes. The Human Genome Project has really allowed us to study the molecular basis of health and disease, and that's really where it's been very, very useful and continues to be today, because it's a fabulous resource. So genetics as such is the study of single variations, and we'll touch. Kicked off what's known as the omics area, the global looking at whole areas. So genomics is the interaction of our genes in the genome. And it really has started to emphasize our systems biology, which is where we start to move into this interaction of our genetics, our genomics with our systems within our body, and then with ourselves within our environments. So just a brief diagram here to look at this transition from traditional genetics, which is where I really started with my research, and then moving into this genomics area. Then we started to look at our epigenomics, which is our gene expression, proteomics, so how our proteins interact within our cells, our global protein sort of areas. Then our metabolism as such, and our actual cellular activity, moving through to those specific organs and tissues, our systems. And then, of course, as a whole, we are a whole organism, and thinking about that organism as one person. So this is where we're just going to slot into the functional medicine model as such. And it's well recognised that our genetic predisposition is really important when assessing those antecedents, triggers and mediators, which I believe you've had a bit of a flavour about. It's very helpful for a nutritional assessment, or really for looking at the person as a whole, their lifestyle, very, very useful there. And of course, the intervention side. So that's what we can do as practitioners to help. It very much sits alongside the mental, emotional, spiritual influences side, and in between that and the experiences, attitudes and beliefs. So this is a diagram from the Institute of Functional Medicine. They've actually slotted it in there at the bottom, so you can see where that goes. So just a recap of molecular biology. Deoxyribonucleic acid, DNA, is our genetic template. We know that. Most of our DNA is located in the cell nucleus, so we're going to focus on that. There is a tiny amount in the mitochondria, but we don't really have the scope to talk about that today. Transferred from generation to generation, so therefore it's inherited information. And as I said, approximately 3 billion nucleotides, which is quite a lot, really. Our nucleotides are made up of a phosphate group, a ribose group, and nitrogenous base. So you may want to recap on that, the structure of DNA, think back to Watson and Crick and what they discovered. And then we've got our 4 bases. So our 4 bases are adenine A, thymine T, cytosine C, and guanine G. So within the concept of this talk, we're thinking about single nucleic polymorphisms, so commonly referred to as SNPs. And these are basically just a slight change in the genetic code. So our basic genetic code, if we laid it out, is a change in the letters. And so that we have this replacement of one DNA letter basically results in an altered protein. Effectively, that could be it. We'll talk about some of the ins and outs of that, but there is the potential for just a single base change to affect proteins. And then if we think about the omics, how that would affect our metabolism, our overall as an organism. So let's just think about this for a moment. This is one example of the impact of a change in the DNA. So this is a single change here. We have on the left a normal beta globin protein code. So this is would be our gene expression from DNA to protein. So we're talking about transcription, translation. And we can see here there's one change here between that and what's known as the sickle cell beta globulin protein. And that's an A to D change, but it actually has an effect on our protein basic structure. And you have this change from GLUT to VAL there. So that, in effect, changes the whole structure of the red blood cell and then leads to this disease. So you can see how one tiny change can have such a major impact on the body. Now, this is quite an extreme example and is something that we would probably use the terminology as a mutation around. Whereas we're talking about lifestyle effects, we're looking more at a different kind of genetic variant kind of terminology. So we're thinking carefully about our terminology if we're talking to patients. So just to highlight that a bit more, all changes to DNA code are mutations, and that's a given really, but we need to be careful about our terminology. So there is this spectrum of effects. A mutation, the term, is generally associated with changes that severely impact function and result in disease. A genetic variation or gene variation is a terminology that we would associate with changes that have a minor impact on function but still recognise change. And our focus really is on those that can be influenced by lifestyle choices here. And then an allele is the alternative form. So this is the terminology we use for alternative forms of a gene because, of course, we have one from our mother and one from our father. And the allele is the difference that arises by mutation or genetic variant, whichever one we're using. And it's found at the same place on a chromosome and controls the same characteristics. So, for instance, eye colour. You have the allele for blue or you have the allele for brown, etc., etc. So some more concepts here. Just to run through, we have gene. So that's really the terminology we use for a DNA segment that contains all the genetic information required to encode an RNA and protein molecule. So if you've done any molecular biology training, that would be really your transcription translation, that central dogma, or what was known as the central dogma. But we now know there is so much more to the molecular biology than this. Genome is the term given to the complete set of genes for a given species, so we're thinking more as a global term. Our gene expression is the process of that gene transcription to translation, how that actually happens within cells, within the nucleus. And then our genotype is what we refer to as the set of genes in our DNA which is responsible for a particular trait. Whereas our phenotype is the actual observed characteristic, again, our eye colour. So if we had the genotype, say, for blue eyes and brown eyes, our phenotype might be expressed as brown only. So we know that there's some slight differences there, and we just need to make sure our terminology is correct under that. So just a couple more here, and we will be explaining these a little bit more, and I appreciate this can get quite complicated, but again, this is just a flavour for you. So epigenetics is really the heritable changes in gene expression. So if we're thinking about genes, are they active or are they silenced? Are they on or are they off? And it doesn't involve any changes to the underlying DNA sequence. And this is often due to our environment. So we know that our environment affects our gene expression and effectively can help to turn on or off genes, whether appropriately or inappropriately. And the body reacts as a whole global system to our environment the whole time. So epigenetics is a very interesting area, and the term was really first used within developmental biology and has expanded since then. So I want to really highlight to you some important bits about epigenetics. It was about 1940s that it was first termed, and it's really a definition that continues to evolve. So it's known that epigenetic changes may not be permanent loss of information, so there's a constant rearrangement going on here, constant shuffling, and there's a potential reversibility of epigenetic changes is a feature which is really relevant to us, because if we can't change anything, then what's really the point of understanding it and reporting it to a patient or a client? We need to have some actionable action that's important here. So there's three main mechanisms that you may want to look into, and again, I don't really have the scope under this talk to go through these, but there are some webinars that I've done myself which goes into this, or you'll find plenty of information on the internet or in textbooks. Three main mechanisms are histone modification. So histones are really the proteins that the DNA is wrapped around a storage mechanism within the nucleus. We also have DNA methylation, which is where a single methyl group, so a carbon atom attached to 3 hydrogen atoms, is your methyl group. And those attach onto DNA code as such, and they can affect gene expression quite greatly. We know that's a major form of epigenetic modification. And also we have what's now known as non-coding RNAs, which originally weren't too much research, but now we're finding that these are very, very important. So these are after that transcribed event, we have RNAs produced, and they can actually go back and affect gene expression, which is becoming ever more fascinating. But really, how does this affect us? What can we do? What is epigenetics in action? So it's really this understanding that we have our genetic predisposition, so that's our genes as such, our set that we inherit, and combined with our environmental factors can equal either function or dysfunction. So it's understanding that our inputs really have an effect on our genetics as such, and our body acts as this integrated system which produces what we've called here the response, but effectively is changes in our DNA, our gene expression, the phenotype that comes out really, and it is very much a complex set of small molecules. So the key message here is, if you want to take something away from this slide, is that genes are really not our destiny. Originally, genetics could be thought of quite a scary thing, but now I think we need to think of it as something very modifiable, and that we can work alongside our genetics and our epigenetics rather than battling against them. And so another key message is really that dietary and lifestyle choices really have very strong influences and very, very useful, and something that we should be thinking about. So genetic variation again, we know that the DNA code are genetic variants, but there's this spectrum of effects on terminology we spoke about. So when we talk about DNA polymorphism, that's the term used when a sequence variation occurs in a human population with a frequency greater than 0.01. So there's kind of a cutoff level where we think of something as very rare or something that's relevant to populations, and that's where we're looking really at population level. And the Human Genome Project gave us massive information on this and identified over 3 million base substitutions. So that's that 0.1 to 0.3% of us that's different from anyone else. And it also gave us this allelic frequency, and those have become known as single nucleotide polymorphisms, or SNPs. So most SNPs are really synonymous, which means silent, which means that they don't really do very much and they just sit there. But there is this 3 to 5%. So we're talking about 3 to 5% of these 3 million changes are functional, what's known as functional, and they can induce changes either to the protein or cause the protein to be nonsense, or they can influence how the gene expresses itself. So there's a wide spectrum of effects there as well. So really we've cut down here again. We've gone from 3 million base substitutions, and then we're now looking at 3 to 5% of those, and we're really focusing on those, and they do have an effect on our phenotype, so what we express, and that's the difference between us all. So we have what are known as SNP association studies. So these are GWAS studies, and these are where we look at why some individuals are more susceptible to what we call non-communicable diseases such as hypertension and type 2 diabetes. So these are studies that take one population and look at their SNPs, their differences, and compare to another. So they may take a group with hypertension and compare to a group that doesn't have hypertension and look at these functional SNPs within that and try and pull out ones that are very relevant to that. So what we do as a company is we look at smaller scale mutations. So we're very keen on calling these genetic variations, and we group these into different classes. So we have base substitutions, so that traditional SNP. We have what's known as insertions, where some nucleotides are added into a sequence, or deletions, where some are removed from a sequence. And these really, we focus really on the SNPs, the base substitutions. This year, we're looking at a range of topics. One that will be particularly of interest to you, I'm sure, is the dental health project we have going on. So hopefully at some point, we'll be able to give you an update on that, where we're looking specifically into SNPs that are involved in dental health. We're also touching on sports performance. We have a project going on sports performance, and we're looking at histamine intolerance as well. What we don't do as a company is look at large-scale mutations. So these are abnormalities in the chromosomes that severely impair function. So if we think back to that sickle cell disease that I spoke about, and thalassemia or cancers, we don't look at that area. We're entirely lifestyle-based changes. So back to this epigenetics is this major source of genetic variation, and we touched on the 3 different mechanisms that were there. We looked at histones, we've talked about DNA methylation, and then those RNA molecules. But all cells within an organism contain the same genetic material. We know that, that's our code as such, but they have different gene expression. And epigenetics helps us understand how one cell will express differently to another. So a bone cell is quite different to a skin cell, or a brain cell is quite different to a skin cell. And epigenetic describes the phenomenon in which genetically identical cells or organisms express their genomes differently, causing these phenotypics of what we observe differences. So we've got a little diagram here to help you understand that. When we have cells that are genetically identical, effectively clones, but then within the organism, they begin to become different, phenotypically different, and this is down in part to our epigenetic mechanisms. So this is all about our individuality, how our epigenetics affects us and what makes us us, which is really important. So some studies have established phenotypic changes in response to environmental influences are really based on genetics and epigenetics. And that's something that's very much come out of the Human Genome Project, is understanding that it's not just down to the code that we inherit, it's down to our environment and our epigenetics. And obviously diet is one of our key environmental influences. So just a few examples here of things that can affect our epigenetic mechanisms. So various foods, sulforaphane is known to be a histone modifier, as is anarchaic acid, which is from cashew nuts. So these affect those histones. Then we have curcumin, which is found in turmeric, which can affect those small RNA molecules. And even lycopene, which is found in tomatoes, especially when they've been cooked, is known to regulate methylation. So the food that we eat has a direct effect on our gene expression, our epigenetics. And really, this was famously identified in some studies, some laboratory studies that looked at mice, and these are known as agouti mice. So they have really shown the importance of DNA methylation. So that's one of those epigenetic modification that we looked at. That's that addition of the carbon and 3 hydrogen onto the DNA code. And all mammals have a gene that's known as agouti, and it's briefly expressed in development. So it's a developmental biology gene, but it then remains silenced due to methylation for the remainder of life. So its function is very much in the development phase. But when a mouse agouti gene becomes unmethylated, its coat, the mouse's coat, changes. It becomes yellow. So you can see here on the right, we have a brown mouse and a yellow mouse. And this is just a change, a reduction in methylation of that agouti gene has caused that change. And also the mouse is obese. It's put on weight. And then the further studies found that they were more prone to diabetes and cancer. So having less methyl groups attached to this gene has a huge effect on this mouse. And when the mouse agouti gene is methylated again, the coat becomes brown and the mouse has low risk of disease. So that wouldn't happen in that gene, in that mouse, but it may happen in the future. But that's all down to the diet that the mouse is fed. And as this is an epigenetic change, it's passed on to the next generation. So if that mouse went on and had another litter, those mouse would have yellow coats and be obese. But we've found through research that this hypomethylation is reversible, which is really important for us to know, so that we can affect our epigenetics and affect our well-being through our lifestyle choices. So this is becoming very much more a topic of conversation in the general health, public health sphere of knowledge, really. I think the public are beginning to understand this much more. So really about this importance of environmental and lifestyle epigenetic modification, but there is a lot of information out there, and it could be quite difficult for people to understand what's right and what's not. So you will often get lots of headlines in the news, and they're very contradictory. One day it'll be one thing, one day it'll be another, and it can be very confusing for the public to understand. And that's really where we come in, is to try and sort out all this information for them and personalize it to themselves. So the research is ongoing around these epigenetic changes. I think we're far off from knowing the definitive answer, but there's much more information coming out that's very relevant to people and is actionable, things that we can do easily within our lives. And these are induced through lifestyle choices. So this diagram here was from Harvard Magazine in 2017, and they've picked up on some of the main ones here, like fast foods or the medicines, how you smoke, what exercise you do, etc. What I really want to highlight is that epigenetics is a term that's evolved, and really it has two key components. So we're looking at the response of our system to environmental influences. So that is the environment and lifestyle that we choose to live has effect on our epigenetics, and has become known as our epigenetics. But also is this memory system inheritability of epigenetic markers, as we saw in the agouti experiments. So there's two mechanisms to think about going on, but crucially we can affect both of them. So it's clear that epigenetics is a crucial factor in the maintenance of our whole system and our balance as such. So this is just a quick slide from a study from 2005, actually, which looked at the actual DNA chromosomes of twins, so identical twins, and they were looking for methylation changes within those chromosomes. So it's just some example here for you. If we see on the left, we have the chromosomes of two 3-year-old twins side by side, and essentially if we look at the pattern of methylation, this is quite a crude diagram, but it shows us nicely in a nice visual way what we can understand, that they look very similar. The patches of methylation are being tagged and highlighted there, and they look very much the same. If we look on the right, we have a pair of 50-year-old twins, again identical, but we can see that there's much differences in the colours if we look at a basic level of the inheritability has changed effectively, and this would be from their lifestyle. So their inheritance of their epigenetic factors would have been the same, but as they've gone through life, they've diverged due to their lifestyle modifications. And I think this is a really nice representation of that and allows us to see these two mechanisms very much at work. So the message really is that traditional nutritional advice is very much based on the average and doesn't take into account individuals. So if we think about some of the advice that's given out to whole populations, so such as vitamin C research, that looks at the average but doesn't really relate to an individual as such. And so the average person was invented by statisticians, not by biological evolution. So that's something to bear in mind when we're thinking about the amounts that we recommend to people and tailoring diet and lifestyle nutrition advice. So I want to move on to really looking at this a bit further and thinking what we can do. What would be precision nutrition? What can we do? So we've got conventional nutrition here, which is those general guidelines based on epidemiological factors and populations. So looking at whole populations here, and that's really public health. And then you do get some personalised individual nutrition. So people, for instance, with hypertension may be put on the DASH diet, cardiometabolic, maybe put on a Mediterranean diet. There's some diets that have been devised for specific diseases and disease states or symptoms. And also functional foods. So those cholesterol lowering foods may be recommended for those who have high lipid levels. If we move on, then we'd start to look at our genotype nutrition. So really our individual genetic variations, looking at our SNPs and our actual code there. So that's where they bring in things like that, those population studies that I spoke of, and looking at the differences in SNPs between people. And what we like to do is really a crossover between this, is that we look at the genotype nutrition and then we go back and try and personalise and individualise it to the person. So bringing a wealth of knowledge together for valuable information for the client or the patient. So really, it's a field of science that aims to understand how diet lifestyle factors interact with our genetics or our genome to produce that phenotypic variation. And it's made up of two disciplines, really. So we've got nutritional genomics as a whole, and then we have nutrigenetics, which investigates our genetics on a dietary response by focusing on our genetic variants. These are those SNPs. Or we have nutrigenomics, which investigates the role of diet on DNA and gene expression. And this often includes that effect of lifestyle choices on DNA structure, and that's known as that epigenetics. So it's really understanding the terminology can be a little bit difficult here. But both of them aim to understand nutrient-gene interactions, and this is what we know as nutritional genomics. We just put this into a diagram that might help us to understand it a little bit more. So if we think about the classic research on nutrition, really considered the effects of macronutrients and then micronutrients and determining the physiological requirement for these. The primary objective of this was really to prevent nutrient deficiency, such as that scurvy and vitamin C. So looking at vitamin C levels when we were looking at the population. But this implies that all people will benefit from identical diets, and that people have no different nutritional phenotype, which we now know is not the case. Whereas nutritional genomics is that relationship of individual genetic variations and their individual requirement for nutrients. So we've got nutrigenomics, which describes how diet modulates the expression of genes, and it also looks at high throughput screening of genomics from nutrition. And then we have nutrigenetics, which describes how human genetic variation results in distinct nutritional requirements. So we really need to make sure that we look not only at the relationship between food and specific gene variants, but also how foods can influence the expression of genes and protein. And that's the key mission here, is that really epigenetics is very, very important too. So we think about a nutritional genomics assessment. What can that actually do for our clients? So if we little diagram here that's expanding on the last one, it really can provide an in-depth look at a complex case. So it can be very, very useful, as is functional medicine, and they sit alongside each other and can help you look from a different perspective. It looks at the DNA, obviously, and transcription and translational level. And it can lead to better compliance from patients and clients due to really increased understanding. So it's all about knowledge and getting them to understand and take steps for their own health. And it can be very, very motivating to understand your genomics as such. And when personalised with lifestyle factors, this can be a very, very powerful approach and very useful. So really, if we think about the challenges facing healthcare today, it is about personalisation. How do we move beyond this assembly line medicine, this sort of whole population look at treating and treat those individuals? That's a huge challenge for the NHS in this country, in the UK, huge challenge for all areas of healthcare, affecting yourselves as dentists right through to nutritionists, GPs, doctors, nurses. It really is a huge challenge. And also we need to think about prevention. How do we prevent illness before it occurs? What can we do? Can we learn something from our genetic variations or epigenetics that might help prevention or future generations? And again, the Human Genome Project really showed us that our phenotype results from our genes and from our environment. So in practice, we need to understand the genetic association with human biochemical function and the connection that that has with nutrition and lifestyle, and then how we can offer clinical tools with which to practice, which seems like a huge scope, but actually we've broken it down for you to try and make it as easy as possible to give you as much time really with the patient or client, rather than having to wade through mountains of genetic information. The information is more accessible and available for you so that you can get straight to helping. So we've got on the left here, nutrigenomics, our dietary inputs, and our nutrigenetics. We look at both. So those gene variants, those SNPs. And then we're considering how they act together to affect gene regulation, the transcription, translation, and metabolism. So that's that central dogma, which results in a phenotype. And then obviously we know that the changes that we have in our life can affect our state of wellness. So it could lead to disease, wellness, what is our actual characteristic that is presenting at the time. So the myDNAhealth approach really aims to understand all of these areas together and help you with lots of information for both yourself and your client or patient. So it starts with a full analysis of the symptoms, and there's extensive questionnaires for patients and clients online. It's a very useful portal, and also takes into account their genetics. So it's a simple cheek swab that will be undertaken to start to put these two areas together. Out of this comes an extensive report where they look at risk scores in over 27 areas of health. I believe it's more than that now, and what this result means. So what is actually to be done, what can be done, and why it matters. So there's a wealth of information here. It's an extensive report, and you can customize it to your client or patient as much as you like. You can remove some areas and focus on one. It's entirely personable again. Then we have a lifestyle prescription, effectively. What can we do? And there's lots of information and advice within the report, and also within practitioner notes that don't have to be passed on to the client or patient, which really helps lay out lifestyle changes, foods, nutrients, supplements, as far as you'd like to go, really. The nice thing about this system is that you can then continue to track your patient or client, so they can retake the questionnaires, which is effectively the epigenetic side, their lifestyle, and that changes the report. So you can have this continuum of care, really, and also you could input further results, functional tests, for instance, things that you've observed in clinic, and go back to generating a report which is ongoing and can help with patient care. So I am the scientific officer for myDNAhealth, and I've developed a framework of research. I'm very passionate about making sure that the research is up to date and useful. So when I'm looking at the genetic data or even epigenetic data, I really make sure that there's 3 areas that are very well ticked off. So make sure that first that anything we're reporting and looking at is very relevant, and this is something we go back and review. So we look at population, we look at health and well-being, and all of this is assessed. Also make sure that it's very reliable. There's a good backup of research and functional results. So look at it from all different angles and making sure that it's been very much tested. And then again, if we go on and test in our test, make sure that the physical test that we do within our own lab is very, very good and robust. And finally, how to make sure that anything we do report is actionable, to make sure that there's something that can be done, that the research is there in the background to help modulate these lifestyle choices and advice that we're giving, and to really make a difference. And that's the important point to be made here, is that we need to be making a difference. There's no point in testing genomics and not looking at the epigenetics, really. We need to make sure that it's actionable and that a report is able to give some benefit to patient or client. So I really, from this point on, wanted just to look at inflammation. Now, I know that you're interested in inflammation within the context of this conference. So I've just gone through here some of our work that we've done on inflammation. This is based on resources that are available to practitioners and will be available to you if you've signed up for an account. And I believe you still can at any point just go to and log in and create yourself a practitioner account. And there's a wealth of information on there to help you with some tools. So really, we know that our immune system is our body's system of defense. It's highly complex and encompasses a wide variety of cells, antibodies, proteins and chemicals, and also what we call immune-mediated diseases. So really, we've put some information together to help focus on those clients who may have some inflammatory related conditions. So our immune system may be divided into 2 areas, but they're different systems. So we know that the innate immune system is the non-specific immune system, and is really made up of those cytokines and enzymes, and the complement system. And that's our first line of defense. but it doesn't have an immunological memory as such. And then also we have the adaptive immune system, which has that immunological memory, is a specific immune system, and is that longer term response, and involves those B and T cells and antibodies and cytokines. So innate immunity is really our first line of defense, and it's very quick to act. And that's that physical sign of inflammation that we may see if we cut ourselves. So the red swelling effect, that would be our inflammatory response. And it's really that process of inflammation whereby we activate monocytes and macrophages and release what we call pro-inflammatory mediators, such as TNF-alpha and interleukin-6, which I will touch on in a moment, causing inflammation in the tissues. And because our innate immune system is non-specific, we know that healthy tissue can often get caught up in the process. And this is a normal response to a wound, for instance. But when inflammation is kind of rampant throughout the body and becomes chronic due to other mediators or, for instance, stress, it can lead to inflammation within the body and some chronic inflammatory driven diseases, which we have here, or conditions. So there's some laid out for you there. So really, this inflammation is, as I've said, based to acute inflammation and chronic inflammation. So we have acute, really, which would be, for instance, that wound where we have a short cycle and then an end of stimulus, we get a healing process and the inflammatory response is turned off. But within the chronic inflammation, we have this repetitive cycle of ongoing stimulus, which could be anything from hormone imbalance or stress, persistent infection, the food or lifestyle leads to this repetitive cycle of disease. And these environmental triggers are very much important here. And there's where we're starting to tie in our epigenetics, which may produce a pro-inflammatory gene expression environment. So really, we know that these environmental triggers in the form of diet, lifestyle, we've also got here stress, injury, bacterial LPS, our food and our environment may mediate pro-inflammatory gene expression. So these are highly variable and different to everybody, and also modifiable by many different factors, which are including leaky gut, which you may have heard of, and the food and environmental allergens, which activate what's known as nuclear transcription factor kappa B, or NF-kappa B. And NF-kappa B really acts as a facilitator for the amplification of this message of these specific gene products. And in the case of NF-kappa B, most of the genes that appear to be influenced are those really that increase the production of pro-inflammatory mediators. So that's what this diagram is doing, is just working you through these pathway towards chronic inflammation and that repetitive cycle that we spoke about. And some of these mediators include things like CRP, C-reactive protein, via interleukin-6, or R-eicosanoid balance via essential fatty acids, things like nitrogen oxide, collagenases and matrix metalloproteinase, TNF, etc., reactive oxygen species, and oxidative stress. So that's that whole package of information there. And obviously, this is a vast area and really can be difficult to pinpoint what's going on with your patient or client. So what we've done is trying to come up with a framework that makes these areas that may be triggering and mediating this response, and allow you to think about these areas and to effectively focus on these and calm the area down, which may lead to some resolution or help or improvement within your patient or client. So under our inflammatory risk assessment framework, we have some areas to think about here: stress, trauma and sleep, diet, infection and dysbiosis, atopic disorders, so an IgE response there, our xenobiotic toxicity, so really our environment, and I know you're very much also interested in detoxification, so that would come under that area. Our endocrine balance, so our hormones very affect our inflammation, our essential fatty acid balance, and of course our genetic mediators. So really I'm just going to run through a quick look at this framework and how this can help you in this specific case. So if we first look at genetic mediators, so we're looking really at that nutrigenetics, how is our actual gene variants affect our inflammatory response? So a couple of classics here, we've got our IL6R, which is receptors, so IL6R receptor gene variant and TNF alpha. And I'm just going to talk you through those quickly so that you can understand really a couple of mechanisms. These are just examples, and we'll give you a flavour of the type of research that we've done and the areas that we look at. So interleukin 6, this is our first mechanism. It's a interleukin 6 receptor. It's a complex, so interleukin 6 is a complex cytokine, and it really has a very critical role in the regulation of that inflammatory response, really very much involved in a pro-inflammatory effect. And we touched on it briefly before, and it comes under that mechanism. But genetic variation in the interleukin 6 receptor gene has been associated with some risk of several of those immune-mediated inflammatory diseases, such as coronary heart disease and rheumatoid arthritis, so those conditions that we spoke about. And really the functional mechanism of IL-6 works through the balance between two forms of the interleukin 6 receptor. And there's two forms. There's a soluble free version and also a membrane-bound version. So I'll just talk you through those two versions and how a genetic variant, that one change, has made a huge difference to this receptor. So as I said before, IL-6 has these inflammatory actions through the membrane-bound receptor and a soluble receptor. And it's really the balance of these two that can lead to our levels of inflammation. Not exclusively, but can have an impact on the system. So really, classic interleukin 6 signaling forms part of a tightly regulated acute immune response, and that's what we talked about under the conditions of wound healing, for instance, and functions through a membrane-bound form of its receptors, which is found only on the cell surface of hepatocytes, so liver cells, and immune cells such as leukocytes. And we can see here 6 in the blue triangle to the membrane-bound IL-6 receptor, which has been lined here in orange, and causes what's known as homodimerization, which is a joining together of two proteins with its co-receptor, which is known as GP130, and we can see that here in green. So I've just done a basic diagram for you to visualize what's going on. And this results in what's known as a phosphorylation event, which is signaling response, and initiation of a cell inflammatory response, basically. And this message, which is really restricted, if we think about it, to the liver and certain immune cells, so leukocytes and macrophages. This response is very tightly regulated and is very much a functional response and can be very, very appropriate. But alternatively, we have this truncated form of the IL-6, which is free from the cell membranes, is able to bind to IL-6 and initiates what's known of as trans signaling. So the soluble of the IL-6 receptor is produced by proteases, which effectively are there to cut the receptor free of the cell membrane. The IL-6 receptor becomes smaller, truncated, and is freed from those hepatocytes and leukocytes, and then circulates throughout the circulation. Then the IL-6 and soluble IL-6 receptor complex is able to stimulate any cells which express GP130. So that's that co-receptor there, which also leads to activation of the cell signaling response and induction of that inflammatory response there. So we can see that this is a bit more whole body going on here. Now, we don't undergo systemic inflammatory responses on a routine basis. So every time that this would be cut free of the membrane, we would undergo an inflammatory response, and that doesn't happen. And we did a bit of research around it. We found that this mechanism has a clever regulatory aspect to make sure that it doesn't overwhelm us. So IL-6 stimulating a inflammatory response every time that happens would be undesirable. But this response is in effect buffered by a soluble form of GP130. So again, GP130 becomes solubilized and is snicked off the membrane. And we have this another protease enzyme which cuts it free from the surface membrane. So what happens in effect is soluble GP130 is enough to easily buffer the majority of this chronic inflammatory response. And only when an organism goes into really a severe immune-mediated event, such as an infection or chronic underlying inflammation that's overwhelming, this would be enough not to buffer IL6 whole system inflammatory response. So really what this IL6, we look on the right, the IL6, it's receptor soluble and co-receptor GP130 as a whole, when they're bound together like this, are very inactive and just floating around in the system. Once that GP130 is effectively used up and completely bound, that's when we would have activation of cell signaling going on, activation of some immune response due to the overspill. And really that research points to us understanding that there's a genetic variant which confers some changes in the chronic inflammatory conditions. So really, if we think about it, I've just laid out here what a single nucleotide polymorphism can do. There's a functional SNP, which is within the part of the receptor protein, the IL6 receptor protein, which is found within the site that is snicked off from the membrane. So it changes the way that the receptor is removed from the membrane. So it's effectively within this cleavage site of IL6R, the receptor. And what it does is having that genetic variant actually causes an increased cleavage rate and more of the soluble interleukin-6 receptor. And as we saw in the last diagram, having more soluble interleukin-6 receptor may lead to more whole body inflammation, effectively also if it becomes overwhelmed by a lack of GP130. So it becomes it overwhelms the system. And population studies show that this genetic variant protects against chronic inflammatory conditions such as listed below. So we've got chronic heart disease, rheumatoid arthritis, but also increases susceptibility to asthma. And this is thought to be due to an increased Th2 cell recruitment, if you want to look into the technical side of that. So really this is a direct nutrigenetic effect, and depending on which genetic variant you may have would affect your inflammatory response to some degree. So I've laid out a table here of some of the observed effect of the C allele, which is one of the alleles, so one of the different changes, and it can lead to an increase in IL6 levels, and this is thought to be an indirect effect due to reduced clearance through the liver, and that excess IL6 is buffered by the soluble GP130 and does not influence inflammation, remember. It also leads to a recognised decrease of CRP, so C-reactive protein, and fibrinogen levels, so it reduces that classic signalling as well. And reduces the CRP load and fibrinogen load from the liver, and it decreases the cell surface expression of interleukin-6 receptor and really enhances the effectiveness of the shedding of the IL6 receptor. So that is one example. I just want to move on to a second one. I know that we've been getting quite technical here, so I'm just going to do one other briefly. We're going to move on to another gene variant that's quite effective and interesting, known as tumor necrosis factor alpha. And we're going to consider this variant really quite carefully, but sometimes it's related to regulation of blood sugars, where traditionally has been looked at, but looks at whole inflammatory effect as well. And so many genes have been really linked to metabolism, and studies have indicated that TNF-alpha, which is also a cytokine release from macrophages during inflammation, has an important role in our postprandial glucose and lipid levels in our blood. And there's a genetic variant located in the promoter region of TNF-alpha gene, which is really linked with insulin resistance, and it's known as TNF-alpha cytokine, really inhibits insulin. So what we need to think of here is the different SNP and the effect that that's having within the effect of this actual gene and its gene expression. So it's laid out here, some of the effects. It's a macrophage-derived, as I said, pro-inflammatory mediator. There's a genetic variant of the TNF-alpha gene promoter. So that's the region that drives the transcription of the gene. And it is associated with a direct increase in the production of cytokines. So if you have that particular genetic variant, you are more prone to produce TNF-alpha. And so this is the A allele. And you can see here's a table that outlines it for you. It really increases the risk of producing excess TNF-alpha, which, as we saw, is an inflammatory mediator. And therefore, we have potential increased CRP levels. So really, the advice around this has traditionally been to maintain low sugar consumption, so there isn't a spiking of effect there, because we know about these postprandial events. And there's also research that points to nutritional factors such as omega-3s, those essential fatty acids, things like curcumin and green tea extracts, which can be involved in directly downregulating the risk variants. And there's some very interesting research around that. Because TNF-alpha promotes Th17 induction, which indirectly promotes TNF-alpha and NFKB production, which can lead to this vicious cycle of inflammation that we talked about before. So this diagram is really just to outline this in a different way, because I know it can be quite technical. Just want to remind you about the mechanism of gene expression. So we have here a TNF-alpha gene and its promoter ahead. We have the G allele here. So this is what's known as the wild type, the more normal variety or genetic variant. And so in this case, we're looking in promoter region. Then we have the variant, that A allele, which may lead to increased gene expression of TNF-alpha. So we have more TNF-alpha in the system if we have that genetic variant. And then we talked about this feedback loop that happens from TNF-alpha, which goes and regulates itself, Th17 cells, which in effect then regulates TNF-alpha itself and leads to more production of TNF-alpha. So we have this chronic cycling effect going on. So really it's important to think about those two genetic variants, and that's something that would be done under the report, and there would be advice and lifestyle information there about what could be done to help mediate and improve those genetic variant sort of risk factors. We can't change the genetics as such, but we can change the epigenetics to try and change the expression of them. So then moving on round our inflammatory framework, the next area to consider is really our essential fatty acid balance. So these are these essential fatty acid metabolism pathways, which are known very much to directly affect our inflammatory state. So the pathways of the polyunsaturated fatty acids, which are also known as omega-3 and omega-6, are really very important for inflammation, and they differ. Omega-3 and 6 differ greatly due to some of their biological and clinical consequences. So if we just look at the next diagram, so we have here alpha-linoleic acid, which is omega-3, and linoleic acid, which is omega-6. So we know that on the whole, omega-6 arachidonic acid derived eicosanoids, so these are short hormones that are produced from these essential fatty acids from the cell membrane, have strong and pro-inflammatory effects, while those eicosanoids that are derived from omega-3 PUFAs, such as EPA and DHA, which you may have heard of, have very much more anti-inflammatory actions. And it's the body's ability to convert these essential fatty fats, omega-3 or omega-6, into longer polyunsaturated fatty acids, which affect our PUFA profile and our inflammatory state. So, of course, there's more information on this on the practitioner portal. It's in the report. It would be taught through, but really, it's important to remember that both the omega-3 and 6 pathways compete for the same set of enzymes, which are known as desaturasers. So if we think about what essential fatty acids do and how important they are to health, They're very important in different areas, and we're beginning to really understand that. Not only can they be used for energy metabolism, but they form part of the cell membrane, which affects the fluidity and the permeability of the membrane, and also all of those membrane-associated proteins. They also have direct effects on gene expression, transcription factors, and how they affect our research there. And these messengers, these short-acting hormones known as eicosanoids, which really have the bulk of the pro-inflammatory or anti-inflammatory response. So we've got here the essential fatty acid pathways, which I spoke about before, and we've got the omega-6, omega-3 moving down. And as I said, these are key pathways that are known to have profound effects on our PUFA profile and our inflammatory mediators. So if we run down, we have omega-6 on the left, which tends to be more pro-inflammatory, omega-3 on the right, which tends to have more of an anti-inflammatory. So these eicosanoids, these short-acting hormones, we really have a balance within our body, which affects our inflammatory response. So we know that alterations of two specific desaturase enzymes from their genes, so the enzymes are known as delta-6 desaturase and delta-5 desaturase, and the genes are known as FADS1 and FADS2, are associated with inflammation. We have two SNPs that we look at, one within the D6D, so the FADS2, we know increases activity of that particular enzyme and would lead to a predisposition of running down the omega-6 side, so pro-inflammatory, whereas a decreased activity of the D5D enzyme, that FADS1 gene, there's a SNP there that can lead to decreased activity, and so less omega-3, less of the anti-inflammatory response. So it can be very useful to know which of these SNPs you may have, and your sort of which way your pathway, your essential fatty acid pathway, may follow. So there's some highlights here for you, and again, this would be in the report and it would talk you through it. But we know that PUFA levels are really influenced by genetic availability. So we know that our genetics are very, very important, and also we know that from an epigenetic side, what we're putting in. So if we consume oily fish, for instance, vegetarians, they don't consume oily fish, so it needs to take in different forms, and that our genetics can affect our ability to process these particular essential fatty acids. So the FADS1 SNP is associated with that reduced D5D activity and that impaction of omega-3 precursors. And the FADS2 SNP is associated with an increased D6D activity, and that really impacts downstream production of the omega-6 precursors. So from an intervention point of view, if we think about what may pop up, weekly portion of non-farmed oily fish, so wild type salmon, for example, or consider supplementing EPA or DHA if a person's not compliant to eating enough fish, or we could reduce their omega-6 intake, so looking at certain meat and poultry that are particularly being fed a grain-based diet and trying to reduce that omega-6 side, or eggs. Non-compliance consequences, well, really the omega-6 3-6 ratio balance is known to be linked to chronic inflammation through those mechanisms that we spoke about before, so the inappropriate cell membrane fluidity, increased inflammatory eicosanoids, those short-acting hormones. So really our genetic variants and our lifestyle have very much an effect on our inflammatory balance. So I just want to move on to and briefly go through the rest of the areas of this this framework. So of course we know that hormones affect and are affected by inflammatory status, so that would be one area to consider. I've listed here some hormones and kind of maybe low cortisol, DHA and testosterone, high estrogen, and prolactin can be indicative of inflammatory response. You could also use some of the resources that are available to you within the practitioner portal on the report. You could look at their stress questionnaire, so what's going on there. You could look at their sugar questionnaire, what are they intaking, what foods are they eating, and how's that affecting their hormone balance. Things such as insulin, we know, varied by the foods that we're intaking. Also, we have questionnaires on estrogen, and also we're looking at SNPs in this area, so genetic variants, which would impact some of these hormones. So we just probiotic toxicity. This is, as you know, exposure to foreign substances. And of course, I'm sure you're very interested in the measure would be something that we would assess. So we know that accumulation of toxins can produce effects on the whole body, endocrine disruption, leading to oxidative stress, and then crucially that inflammatory pathway. And also now linked to mitochondrial dysfunction, so changes in our energy levels, and that's related to conditions. So under this, we would consider our whole detoxification panel, which unfortunately we just don't have the scope to go through today, with information online. And within this, we would look at our liver questionnaire as well as some genetic variants, understand what the client patient's doing within their life, and really assess their xenobiotic levels. To just pop two together here, so diet and those atopic disorders, IgE mechanism, because we know that food intake and nutritional status very much affects inflammation. And again, a huge area that we could talk about in another webinar. But really, we know that macronutrient balance, such as things like obesity, food balance, food allergies and intolerance, nutrient deficiencies and prebiotics, all of these areas we would consider. And maybe under our framework, we would look at our cravings panel. There's genetic variants that show us maybe we're more likely to crave. Our sugar and fat metabolism panels, so SNPs, and questions that help us understand how we are processing both of these macronutrients, and also antioxidants. There's SNPs that are very much related to maybe we need one specific antioxidant more than another, and looking at the client and patient's diet to really understand their actual need. So we then could move on to infection into this. We know that chronic exposure to microbial factors mediates inflammation. By that, we're really talking about desirable microbe, or what has been termed sometimes as bad microbiota or bad bacteria. We don't really like to use that terminology, so we're really looking at those that maybe mediate inflammation. But dysbiosis can be all-encompassing, all of the different areas of the body, and one key one, of course, is orodental. So dysbiosis infection can start in the mouth, that can have a direct effect on our chronic inflammation. Just consider gut permeability, so that's what we really would call leaky gut. Very much know that if the gut is leaky, if there's holes within the enterocyte membrane as such, this can be associated with chronic inflammatory parts, foodstuffs and others. Bacteria are crossing the digestive tract, going into the system and influencing our inflammatory response. Our immune system has to recognize and deal with these, and that can lead to that chronic triggering of the response. So under this, we may consider the gluten sensitivity panel, which is known to, gluten is known to affect those tight junctions between enterocytes and can lead to some leaky gut. Now, it's very much in the public mindset at the moment is stress, trauma, and sleep. So we're beginning to really understand how our psychoneuroimmunology is much affecting our immune response. So we can think about something stressful and increase our pro-inflammatory mediators just through that. So it's very important to a patient lifestyle through their stress questionnaire. You can use that resource, very useful. The burnout questionnaire, again, there's loads of resources here. You can look at their cortisol release through questionnaire and how they're sleeping. And these, again, can be completed continuously. You can look back in 6 months and change, and that would change your report. Some very useful areas to think about when we're looking about chronic underlying inflammation. So that was a very quick run through of our inflammatory risk analysis framework, just to really give you a taster and make you think about some of the different areas that might be involved in inflammation, how we can look at that from a lifestyle and diet point of view, how we can look at that from a genetics point of view, and really how we can bring that all together to be useful for a client or a patient and is practical and easy to use in clinic. So in summary, we can see that nutritional is very useful. It can really provide an in-depth look at complex cases. It gives you a different perspective, somewhere and a framework where to start from. So both the DNA and the transcription and translational levels. So we're looking at all levels here. And as I said before, it can lead to better compliance standing, both from the practitioner side and from the client-patient side. And really is together and can be very personalised when combined with lifestyle factors. So it's very useful for targeting goals, coming up with an action plan, and really making a difference to your patient or client. So I'd like to thank you if you've got to the end of this, and of course, for any up-to-date references, we're continually updating on our website. There you can go to that site and look for references that are direct and others. And of course, you can sign up for your practitioner account if you already haven't. It's completely free. And I'd like to thank you for your attention. I apologise once again for not being available to talk to you last Saturday.